Hi, everyone, and welcome to the Repligen webinar series. This is our 6th episode of 2022. My name is Dervila Campbell, Marketing Manager at Repligen. Today's webinar is the second in our series focused on upstream process intensification. The title of the webinar is Key Milestones that Define Modern Upstream Intensification Standards. Our presenter today is Melissa Carpio, Director of Global Upstream Field Applications at Repligen. Melissa will review the past, present, and future impact of upstream intensification, including some commentary on emerging applications. Before we begin, though, let's cover a few points on how today's webinar will work. First, the webinar is being recorded so that those who can't join today's live broadcast can view it later. Second, we do have interactivity on the webinar platform. In the panel to the side, there's a single poll question. Please take a few seconds to answer it. We do appreciate the feedback because we use it to guide future content creation efforts. You'll also see a live Q&A panel right next to the poll. You're welcome to submit a question at any time during the webinar. You can use the upvote feature to vote for questions you like so that we know how to prioritize them. And if we don't get to your question today, you'll receive an email response within 24 hours. At this point, I'll hand it over to Melissa for her presentation on key milestones that define modern upstream intensification efforts. Thanks again for joining us today, and we hope you enjoy the discussion.
Thank you, Dervila. As Dervila said, my name is Melissa, and I am proud to be leading the team that has implemented some of the application examples that I'll be sharing with you today. So to get started, I will be going over the history of upstream intensification, and then I'm going to give a technology overview of the Excel ATF, and then I will show the implementation and impact of the Excel ATF. So to get started, process intensification, it's defined as the engineering principle of transforming conventional processes into more economical, productive, and green processes. In the over 20 years that we've been working with customers on upstream intensification, the drivers vary significantly, but the tools and the strategies for intensification are similar. One of the key tools is a cell retention device. And but some of the main drivers can be bucketed into business drivers like smaller plant size, reduced cap and OpEx, and then distributed manufacturing. Some of the process drivers include high cell density, increased productivity, maintenance or better product quality. In addition, improvements like wider process conditions and continuous processing. And lastly, and one that we have seen evolve through the years is the environmental. So reduced energy usage, reduced waste, reduced reagents, and then also a smaller footprint. So as I mentioned, the key strategies and how to implement intensification, that does follow a similar path. And with all of it, cell retention plays a key role. Originally, it's been around since the 1980s, and it was implemented for unstable or liable proteins. What it has shown is that you get increased cell density, you get higher volumetric productivity, and you get faster throughput. So the advances in the technology and also certain industry pressures have moved it from its original application of the unstable or labile proteins to molecules outside of that application. And there's about 20 plus years of literature with the implementation of the ATF specifically as a cell retention technology, showing that more and more molecules were being used with that as a cell retention technology. And why? And this is why. So intensified processes can increase productivity by orders of magnitude. And it can be done with diverse modalities and also at multiple points in the workflow. And what I've shown here is three kind of high-level examples, two different cell types, and then also a different modality. So the first example on the upper left is CHO cell growth. And I particularly picked this graph because it does a lot of different things all in one graph. So what you can see here is between day 0 and 4 is normal cell growth. And then the cells get to about 20 million, and they're able to show continuous, maintaining 20 million continuously for about 10 days. And then the next thing that was done was to push it even higher. So it got to between 100 and 120 million. And that was done, for another 10 days. And then last part of it was really pushing, how high can these cells actually go? And that's where greater than 200 million was achieved. So like I said before, the drivers are different. So some people want to keep it, 50 million. Some people want to push it a little higher. And some people, those that do concentrated fed batch, will get it to go as high as possible. That can all be achieved. And that's all shown here. The bottom left is showing SF9 cell growth. And again, this is mostly just showing how high can it go. And in some cases, it got as high as 200 million. In some cases, it even got as high as 350 million. The key takeaway here is this particular example was looking at different bioreactors. And that is the nice thing about the Excel ATF, is that it is bioreactor agnostic. And we've been able to connect to almost any commercially available bioreactor out there. And then the last example on this slide is on the right side, and it's showing vaccine manufacturing. And this is showing both cell growth improvement and also viral harvest improvement. And in both cases, we saw a 5x improvement versus batch for cell density at time of infection, and then also a 5x improvement of viral yield at harvest. So as I mentioned before, the Excel ATF is the cell retention device. It does interact and interface with other components of the upstream process. Here, it just shows a schematic of a typical layout where you have the Excel ATF device, and then you also have the controller. It can be connected to any bioreactor, and then it also can be connected to any type of collection vessel. And then the technology itself, I'm sure most of you are familiar, but for those who aren't, I just wanted to give a brief overview of how the ATF works and how it's different from other cell retention technologies. So it's a diaphragm-based pump. So there's an air cycle and a vacuum cycle. So the vacuum cycle will pull the cells from the bioreactor through the filter, and then the air cycle will push it back in. And while it's doing that, two things are happening. One is that you can do a cell-free harvest into a collection bag. And then also, it's because it's back and forth flow, there is a backflush action that actually cleans the filter. And this is important because the feedback that we've received is the ATF, in most cases, is superior to other technologies because it does, quote unquote, clean the filter. And the byproduct of that is that you have longer filter longevity. Also, product yield going through is higher. And in a lot of cases, those two alone, even if it's a 5 to 10% increase in product yield, or you're able to keep the filter from fouling for an extra day to weeks, it does impact those drivers that I mentioned earlier. So when picking a cell retention device, there's longevity, there's product permittivity, and there's also scalability. So this slide, I just wanted to show, again, with the longevity that we've had the Excel ATF products for, it operates as low as half a liter, and we've implemented as high as 5,000 liters in various product life cycle, from process development to large scale development, all the way through clinical and commercial manufacturing. And just a quick kind of data set here showing bench versus a 50 liter, and then also showing 2 50-liter runs to show reproducibility and then also comparability between the bench scale and larger scales. So my last slide on the topic of scalability is just showing the range. So the Excel ATF devices range from what we call Excel ATF 1 all the way to Excel ATF 10. And I'm not going to get into the parameters. Those are available on the website, or you can reach out if you'd like to find out more information about any of these. But I think the key takeaways for me for this is that regardless of the scale you're working with, there is a device that can be used. In most cases, they're available in single use or stainless steel. And they come in a variety of different pore sizes. The other important aspect, and as these were designed, is that the lumen that's within each of these for the most part, are the same size and diameter. And what happens and what that means is that what you're developing using the ATF 1 or 2 at the small scale, that's linearly scaled up when you get to the ATF 6 or 10 if you're operating in the thousands of liters. And what it is the ATF 1 will have a number of the filters with the fixed lumen diameter. We're just going to increase the number of lumens as you go up to the XL ATF 6 or 10 without changing any of the other parameters. So you're able to scale based on flow per fiber, which is great. So you see that there's a new there under two of our devices, one device, which is the XL ATF 1, and then also the new XL Lab controller. So what I wanted to do to close out the device portion of this, or the equipment portion of this, is to really show that the XL ATF has improved continuously through the years to adapt to the changing needs for upstream intensification. And so we've shown just the last 10 years here, starting in 2012, where the ATF was a finalist for the Bioprocess International Technology of the Decade. And then from there, we have 2014, when Repligen acquired the Refine ATF technology. And starting from 2014 onward is continuous and technology leadership. So the ATF was originally stainless steel only, and with the rise and the importance of single use coming into the industry in 2016, the ATF single use devices were launched, starting with a few sizes, and now almost all the sizes have a single use option. In addition to that, during that same time frame, there was a lot of controller optimization done on both the small and large scale controllers, on the algorithms, and really understanding what processes were being used and how to optimize the control to deal with higher cell densities, different cell types, different modalities. In 2018, Repligen acquired Spectrum Labs, which is the hollow fiber technology, and that's really to be able to control the supply chain and to really understand what makes up the filters. And then more recently, in 2021, the XL Lab controller was launched. So that's a new process development size controller. So it will support the half a liter upwards. And what that is able to do is it includes features like permeate pressure, and then also is able to run dual. So to be able to save some lab space, you can run 2 devices on 1 controller. Also with it came the launch of the single-use ATF1, and that was an ask from the industry to be able to do process development at smaller scales. So the ATF1 will allow process development to be done at as little as half a liter. And as a preview into the future, we'll be launching the large-scale XL controller, and that will take the features that were developed for the XL Lab. So if you have an XL Lab and you're using some of those features like the permeate pressure or the ability to do dual control off of 1 controller, you'll be able to do that at the large scale as well. So this has shown the track record of the device, of the instrumentation. And what I'd like to do for the second half of my talk is really to dive deep into the applications. And this is, as I said before, cell retention, perfusion, it was developed for a very niche market 20 plus years ago. And through the years that we've worked with hundreds of customers with the XL ATF, you can see that the primary application is still CHO, but you can also see that 25 plus different cell types have been successfully intensified. Some of them, would make sense considering the new modalities that are coming up. So HEK293, insect cells, stem cells, but other ones, that over the last couple of years have really piqued our interest, but also shown us where the industry is going. So E. coli for plasmid production, and more recently, adipose tissue, or actually cultured, cultivated meats and seafoods. So that's cell type. The next one is used for, so what process step? And again, this has evolved a lot. over the last 20 years. So the number one application is still perfusion. And that does make sense. But as you can see, we're also getting, the percentages are increasing in other process steps as well, including N-1, the production reactor for cell expansion, and then also concentrated fed batch, media exchange, and a few others. Then lastly, in terms of application, is really what's being made. So what products are being produced. No surprise, the most common is still MABs or MAB-like proteins. But again, you can see there's an increase in virus-like particles, gene therapy, including AAV, recombinant protein enzymes, and our other bucket keeps getting bigger. And that other bucket includes, as I said before, E. coli for plasmids, and then also the cultivated meats and seafoods. But what you can also see here is just the adoption, and us being able to really see how scalable it is, and the impact as more people will scale up and continue to use the ATF technology in their subsequent facilities. So thus far, we have about 50 plus commercial processes globally that use the XCell ATF, almost 100 in late phase, and then hundreds in preclinical phase 1, phase 2, over 200 unique customers, and over 2,000 GMP installations. And the last part that I'd like to share today is examples of applications, both through the years and then more currently. So this slide was developed a few years ago, and it was right around the time where we started to really see people advertising, oh, this is the importance of intensification, and this is the impact that it's had on our company and our facilities and our processes. So Transcenta has said that, with the implementation of the ATF technology, along with automation and connection to downstream, they're able to really increase and make a lot with the facility that they're making with less cost than they were expecting. Sanofi has the integrated continuous facility. WuXi has continued to, this article I believe was from 2016 or 2018, and they've continued even to now to continue to show the importance and the use of continuous, and then also that, what they say in this article is they get 50% of capacity from 5 to 6% of the reactors. So be able to get more from less. And then Samsung adopting their N-1 at 3,000 liters. And the more recent examples are all from this year, from 2022. So just last month, Genentech opened their new clinical supply center. And one of the quotes from the article that they posted that really stuck with me, it was that, 4 decades ago, it was challenging to produce even grams of medicine. And the focus was really scaling up. So huge 25, 30,000 liter reactors. But what they said was the core component for the future of manufacturing is set to play out on a much smaller scale that's modular and more nimble. And really intensification helps with that. WuXi again, this is an article from this year. And what they did was they showed the focus on continuous processing. They also focus here on the need to move to single use. And the last two are specific examples of application. So one is a BMS, and what they're doing is taking what we know already about the benefits of N-1 or any kind of intensification and optimizing it with PAT. I won't steal the thunder too much here because one of our upcoming webinars will focus very specifically on this case study. And then MSD Animal Health, they're showing virus manufacturing. That was actually the case study of 5x cell density improvement and the 5x virus yield improvement. There's a white paper that was out this year, and there was also a recent webinar that focuses on this. So if you're interested, I do encourage you to watch that one. So I'd like to close this webinar by showing you kind of just a summary of how the Excel ATF has been the cornerstone of process intensification and continuous manufacturing for what it's done in the cell retention space. And what we've seen our customers do over the last 20 years is really increase throughput, increase productivity, increase capacity, also be able to be smaller, to be able to build smaller facilities, reduce their reactor size, accelerate their programs, and be able to rethink what facility expansion looks like, more out of existing facilities, or if they do have to expand, do they have to expand as much? So I thank you for your attention, and I'd like to pass it to Dervila for the Q&A session.